Welcome today. Thank you so much for being here again. I think it's been a great first couple of weeks. So we're on week 2 now, and you've already learned a lot. I wanted to introduce Sasha, one of your classmates, and if you don't mind just telling them a little bit about what's going on.
Sure. This is my second year at Cabrillo, and every semester I've done notes for at least one class. I take notes. If you have accommodations registered through ASC, you can go through my ASC into the portal. Somewhere you'll be able to find. I'll upload the notes for anybody that needs them. That's about it. I do handwrite notes, so I'll upload scans, but if that doesn't work, let me know. I'm willing to type them out and upload a document that way. Reach out to me through Canvas inbox is probably the best way. I put my name on a sticky note here that you can find it. If you have any questions or need help getting to the portal, that's about it.
Actually, we can just write your name on the board too, if you don't mind. That'd be great. So Sasha, this is a service for students who have accommodations for note taking. And I so appreciate it that you are doing this. So you do have to be registered through ASC to get access to these notes, but that's really important. So Sasha said you can email her through Canvas. Contact it. Yeah, thank you. And so some of you will have access to it. So thank you again for doing that. I really appreciate it. All right, so hopefully you have had now an opportunity to take care of all of your registration issues. If you have questions, please see me after class. If you haven't been able to add the class properly or get access to the book or whatever. So, a couple of things that I just wanted to make sure that you're aware of. You have assignments that are upcoming. So you may have already completed, you should have already completed the discussion assignment, which is just an introduction to yourself. Hopefully you've done that. The other one that you probably would have done is the Connect. There are a couple of assignments on Connect. So there was a quiz for the first part of the class. And did you have a chance to do that? So great. So that was one. And then there was a lab APR assignment for the tutorial. And now there are other assignments posted which are related to lab. So remember that lab and lecture are different grades. So basically, if you go to the grade book, you will have points for lecture and for lab. And lecture is worth 70% and lab is worth 30%. And so it will calculate a total grade based on the scores in both. So I don't know if you've had a chance to explore that yet. And as you go through, you may have questions about that. And you're absolutely welcome to come and ask. So, so the other question that someone emailed me, which I think is absolutely a reasonable question, is what are we supposed to be doing? I mean, what are we supposed to know? So basically, the lecture exams will be based on material that we cover in lecture. And there are a couple of different places that you really need to get information. The main place is being here in class. Okay, so we go over the topics that we discuss in class are what will be on the exam. I'm not gonna draw random stuff from the book that we don't cover. Does that make sense? So the topics that we cover are outlined in the PowerPoints. So the PowerPoints are really your study guide for the class. Everything that's on the PowerPoints are fair game, is fair game for the test. Is everything that you need to know on the PowerPoints? This is where it gets a little ambiguous. No, it's not, because I cover examples that are not written out. Right, examples, additional information. I may ask questions about that. So it's important that you take notes during the lectures and try to take in what I'm telling you. I am recording the lectures, and those are posted in a module on your Canvas show. Did you see that by any chance? Okay, good. All right. So hopefully things are becoming a little bit clearer. I know it's a lot to juggle with both lab and lecture and assignments and campus and coming to school and, you know, everything. But we will get into the routine and I think you'll figure it out. Yes,
In terms of the students are in the support class, but
yes.
can be geared focused for the lecture or for lab?
The support class, which is on Fridays, some of you are registered for that. How many of you are? So cool. So that class is 2 hour, meets for 2 hours. I was confused last week because I thought it started last week. So some people showed up, which was awesome, but I think that it says in the schedule that it starts this Friday. It really is geared toward lab. So it's an opportunity to have access to the material for another couple of hours a week. But usually I will be there and I'm totally open to reviewing lecture material as well. That said, I'm glad you asked the question because I have to remind you of a couple of things. I am not going to be here on Thursday for a lecture. So we are canceling lecture on Thursday. I have to go out of town for a meeting with another school. So we will do lab. I want you to pay attention to this. I put an announcement on Canvas. Did you see it? So you should be paying attention to announcements. No lecture on Thursday. Lab as usual on Thursday. The Friday class is still meeting. So the support class will meet and Adrian Frisbee is going to be there. So some of you have her for lab. So she's just going to go over the bone material. Okay. And review with you. Okay. So that's that question. Did you, oh, I thought you raised your,
Oh.
you forgot. You just, yeah.
But that's all.
Practicing your joint movements. Oh, maybe. Sorry, yeah.
Is it still possible to join that support class?
It's theoretically cool, but I never know. I'm terrible, as you probably noticed. I like added randomly individuals. So, I don't know. If you really want to come, you can talk to me and we'll see what kind of bribes you want to bring. Yeah.
On the wait list for now.
Then you can definitely add.
Okay.
All right. If you're on the wait list, I will definitely add. Okay. All right. Yes. No, I'm gonna be there. I don't have to leave until after that lab. But unfortunately, I have to miss our lecture, so I have to whatever, why. So are we clear on what's happening this week? Sort of, okay, all right. I'm glad you're clear on what's going on. All right, so we were talking about, we're gonna continue. So this material is now, this is what you're supposed to be studying and reviewing. So I want to get through this lecture and start on cells today. So basically what we were talking about is that your human body is a product of a lot of different evolutionary innovations. And we really need to understand some of these if we're going to understand ourselves. So we are built out of cells. As I mentioned, we are multicellular organisms, so we are more complex. Our cells, we have different types of cells that have different jobs in the body. So our cells have grouped themselves into specialized functions. So groups of cells that carry out a similar function are called tissues. And so tissues are something that we will focus on. There are 4 main tissue types. The tissue types in our cells as vertebrates are epithelial, connective, nervous, and muscular. Okay, so those are the types of tissues. Those are the 4 main categories, and we're going to go through the specific details of those in a future lecture. Okay, but I just want to introduce you to them. So tissues are groups of cells that have specific functions. As multicellular organisms, we are organized in terms of tissues, which are the basic fabric kind of your body. All right. Okay. All right. So this is just a diagram of some of the really grand expanse of time that it has taken to really produce bodies that are as complicated as ours. So the earliest life appeared about how long ago? Do you remember the time? So if we had a timeline, earliest life would be how long ago? Yeah, 4 billion. So these are squiggly little cells. That's supposed to be a cell. I am quite the artist. So yeah, all right. At a certain point in time, a more complicated cell emerged about between 1 and 2 billion years ago. So quite a long period of time went by. And we get a more complicated type of cell, which is called mitochondria. They do have mitochondria. So these are eukaryotic cells. So prokaryotes, or we can say they're eukaryotes. So eukaryotic cells have a true, why did I make this big black dot? Nucleus. A true nucleus, and as one of you mentioned, they have the important mitochondria, which enable us to use oxygen to burn fuel. Okay, so it enables us to produce more effectively what is our energy carrier molecule? Absolutely. So, so mitochondria, a true nucleus that contains the DNA, etc. Okay. So we move forward quite a ways and we get multicellular organisms, such as sponges and etc. So that was a long time ago. And then we have another important event, another milestone. Okay, this is going to be fun. That's a fish. Okay, and this is representing the origin of vertebrates. Okay, which was some 500 million years ago, so about 500 million years ago. All right, so let's talk about that just for a moment. This really is about evolution. Many of you should be familiar or may be familiar with this kind of representation of evolution, which is a tree. So the earliest common ancestor of everything is way back here at the bottom. And that would be that type of cell that lived 4 billion years ago that ultimately gave rise to everything else. So we are on this branch over here with other animals, particularly with primates. So we're way, way, way out here, hanging out on a limb of the tree of life. I love this stuff. So vertebrates really give us a lot of our basic body features. So the evolution of vertebrates, which began with fish some 500 million years ago, presented things like an internal bony skeleton, which you're learning more than you ever dreamed of. That. So we have our internal bony skeleton, specifically that has a backbone or vertebral column that protects the spinal cord, and a skull at the head end that protects the brain. So our skeleton as vertebrates is segmented. It's broken into pieces. Okay? You think about that, we take it so much for granted that we often don't think about the fact that, think about the structure of the ribs, right? They're in a row, they're segmented. Same with your backbone. They're little segments that fit together. We share with other vertebrates genes, which are what? What is a gene? Remember? What is a gene? Right, a gene is a section of DNA that basically codes for a protein. Right? A gene is a section of DNA that codes for a protein. So we as vertebrates all have very similar genes that code for proteins that tell the body how to develop. I mean, just think of that. You start as a single cell, sections of DNA are turned on in that cell that say, you need to have a head end and a tail end. And in that head end, you're going to have a skull. And you're going to have a backbone that protects the developing brain and spinal cord. So vertebrates have genes that give instructions for how to build that body. So there are different kinds of vertebrates besides fish. What are the types of vertebrates? Fish are the most ancient. And then what are some other kinds of vertebrates? Yes, birds. So amphibians, okay, and then reptiles, birds, and mammals. So reptiles gave rise to both birds and mammals. So fish are most ancient, then amphibians, then reptiles are a little bit more recent. Reptiles gave rise to both birds and mammals. But all of these are vertebrates. They have an internal bony skeleton, skull, etc. So mammals give us some specific features. So we share some things with all other mammals. The big deal about mammals is reproduction. They have a really different way of reproducing. Especially mammals like us who are placental mammals. We nourish a developing embryo and fetus in a uterus, an organ in the female, and that during pregnancy, a specific organ develops, which is called the placenta, which enables the little tiny developing beast to be nourished by the mother's blood. Sort of crazy, really. Okay, so you think about it, yeah. They are so weird. They're the egg-laying mammals. So a platypus is in between a reptile and a placental mammal. Because they have a lot of the features that we do. So for example, a platypus produces milk. That's a main thing about being a mammal, is that the female mobilizes the resources from her own body and produces a liquid that then she extrudes and the beast suckles, right? So a platypus also produces milk, but they don't have the same kind of nipples that more advanced mammals have. They just extrude the milk onto their fur and the little beast licks their fur. Yeah, it's like gross. But anyway, it's all kind of gross. But it's interesting that you brought up a platypus, because I was thinking about what is it like for a reptile to reproduce? And you think about these little lizards that we see all over the place. Those are reptiles, right? They're gonna have males and females biologically. The females will lay eggs. And basically, the reptile, like those lizards or snakes, the mom maybe has some parental care, actually more than we have thought about. They've discovered that snakes are a little more complicated in their social relations than we once knew. But basically, a mommy snake can just kind of, scoop out a little dirt or something and then lay some eggs. And then basically she's free to go out and party with her friends. I don't know. Mammals are so different. I mean, think about a deer. The mom has the baby developing inside her body for several months, mobilizing her own resources to nourish it. The baby, in the meantime, is warmed, fed, transported in her body before it's born. After the mammal is born, it is inextricably tied to the mom. The two of them are tied together. The baby cannot survive without her. She's the one who feeds it. A snake, it breaks out of the egg and it immediately has to start finding stuff to eat. So it's ready to go out and find a fly or whatever it is that it's going to. Mammals, oh my goodness, they require so much care, right? So why would you ever do that? Why not be a snake instead of a mammal? Well, if you are a mammal, you have few offspring over your lifetime as a female. The female can only produce a limited number of offspring, but the offspring have a much greater chance of survival. So think about a fish. This fish can have 1,000 eggs inside her. She lays the eggs on the bottom of the stream bed. A male comes by, swims, squirts some sperm on them. They get fertilized. They may grow up, but how many of them are going to survive to adulthood? Not very many. Mammals have very few offspring, but they're nourished, they're cared for, and they survive. So it's a trade-off. What it means for us is that being mammals, we're stuck with this kind of reproduction. Okay. The milk. I mean, they're pretty smart, I think. I love squirrels. In fact, I love anything furry. I'm a mammal freak, so I really do like mammals. So a squirrel has a brain that is as big as a 12-foot long crocodile, which is a reptile, right? So reptiles have very small brains compared to their body size. So mammals have the ability to grow a large brain because they're well nourished in infancy, right? That brain allows them to be smarter, more intelligent, allows them to have more complicated social relations. So mammals are connected socially. They have to be to survive. The female, the mom-infant connection is necessary for survival. So that sets the stage for social relationships throughout their lives. We are a special kind of mammal. We are primates. Primates include prosimians, which are the more ancient types. How many of you have seen lemurs, for example? Like these guys with their long furry tails that have the stripes. They're wonderful. Monkeys are more closely related to us, and apes are the most closely related to us. So we are basically a type of ape. We're very closely related, especially to chimpanzees. So what is the difference between a monkey and an ape? Does anyone know one difference? Or any tail? That's right. Monkeys have tails, apes do not. So apes have bigger brains. They've got other stuff going on. So as primates, we really have taken the mammalian pattern and emphasized it even more. We have really big brains compared to our body size. We require a lot of energy during infancy to grow that brain. So there are longer periods of pregnancy and lactation. Lactation is when you nurse the infant with milk. So primates take forever to grow up. They're very dependent as infants and even in through adolescence. We know this, right? So my kids are in their 20s. Oh my God. Okay, they're still dependent. They would be so mad at me. Primates are super, super, super social. Really big brains, really social. Sometimes people say a lone primate is a dead primate. We rely on our group for survival. And primates have a specific feature that is based on our anatomy that is has practical implications for us. We have an opposable thumb. That means that we can touch our fingertips with our thumb. Now, this may not seem very talented to you, but think about the cat. Like, remember the cat skeleton that you look at in lab? Really different structure than a human hand. What does the human hand allow us to do? Text. Okay. Do you see cows texting? No. So that opposable thumb has enabled humans to change the world. Right? We have changed our environment with our opposable thumbs. We have created AI with our opposable thumbs. So apes are, as I mentioned, our closest living relatives. So their physiology and anatomy is very similar to ours. Chimpanzees share over 99% of their DNA with us. That 1% must make a big difference. I mean, they look different than we do. They're hairy. They don't walk upright. Their brains are not as large. Their brains are about a third of the size of ours. But they're pretty awesome in their own right. Question,
Oh, you see, if the numbers are also very,
bonobos are basically one of the types of chimpanzees. So absolutely. So they're equally closely related to us as common chimpanzees. Yeah, so excellent thing. And give us a lot of insight into it. Do you know, how many of you have heard of bonobos or pygmy chimpanzees? They're pretty awesome. So the our ancestors and ourselves are classified as hominins. Hominins. So that is a term that means modern humans and our bipedal ancestors and relatives. So the earliest human-like things that walked on two legs appeared in Africa. We shared a common ancestor with chimpanzees. So if I were going to draw that tree, I would draw it like this. MYA stands for million years ago, so 5 to 6 million years ago, there was a common ancestor. That means a type of species of ape that gave rise to hominins on one line and chimpanzees, both bonobos and pygmy chimpanzees and common chimpanzees. In fact, they would have split off like that. It doesn't matter. Anyway, chimpanzees and hominins. So what really differentiated this line was that these creatures walked on two legs. So in our earliest ancestors and that lineage were bipedal. There's a lot of evidence for humans that lived in the past or human-like things. So these are some, I don't care that you know this, actually. You don't need to know the details of these specific sites or hominins, but I want you to know that human origins were in Africa. There are many different sites. All of those are indicated with little red dots, where some of the most important fossils have been found that give us really a picture of what happened between here and here. So our earliest ancestors were bipedal, but they still had many ape-like characteristics at this way back in millions of years ago. They had small brains, they had big teeth, really big teeth. I should have brought in some of the skulls that we have. When you go to lab next time, if you remember, look in the cases with the skulls, because we have we have fossil casts that you can look at. It's interesting to compare with our modern selves. So I did want to mention to you that we are the only species of human that are still surviving. I'm going to write this bigger because this is something you should know. That is our scientific name. So we are Homo sapiens. Sometimes we just say we are modern humans. We are the only species who still survive. So these modern humans came from our species, originated in Africa around 200,000 years ago. 200,000 years ago. We're very recent in evolutionary terms. we used to coexist with other human species, such as Neanderthals. So Neanderthals and Homo sapiens lived at the same time. But about between 20 and 30,000 years ago, they became extinct. So we're the ones who are left. It's interesting to think about. All right. So again, Homo sapiens originated in Africa, dispersed throughout the world, including to Europe and the Middle East and other places. Recently came to Europe, even more recently came to the New World. What do we mean by New World? The Americas, right? So North America, Central America, South America. So there's some debate about when people arrived in the New World, but it was after 40,000 years ago. Okay, all right. So we have what is called sometimes a mosaic of features. Mosaic means kind of a cobbled up structure, right? That is existing because of many different innovations at different times. So our large brain is recent innovation. In the last couple of 100,000 years, we have a very large brain. Our short rounded pelvis is similar to those bipedal creatures, the hominins who lived millions of years ago. A lot of these features are associated with what behavior? Pelvis, femur, foot, walking on two legs. We're very odd, okay, as mammals. Most mammals are four-legged, right? They propel themselves with all four limbs. They bear weight with all four limbs. We do not. It's really a weird thing, and it has shaped a lot of our anatomy. So our pelvis is a different shape. Our femurs are long and strong. Our feet are platforms to bear our entire body weight. Our toes are short. And we have an arch in our foot that helps propel us forward. Those are just a few of the anatomical features that we share with creatures from the past. Okay. Questions about that? Yeah.
Would our vocal also be like listed under them?
It'd be a lovely thing to list. There are so many other things you could list, but I love that. So when would the opposable thumb have been? Like when did it come about? With primates. So we share opposable thumbs with other primates. They originated about 65 million years ago, along with a lot of other groups of mammals. So our opposable thumb would date back to about 65 million years ago. Much more ancient than our big brain. Any other questions? Why is this important for you as an anatomy student? This little trot through billions of years of history?
You didn't understand something unless you know that.
I totally agree with that. So that's how I understand our body. We're really, just this weird, it also explains some of our problems. I was just reading an article about how to protect your knees from how many people do you know who have, particularly older people who have problems with their knees, right? Yeah, exactly. And that's because of the way we evolved. We're not perfect. If you were going to design a body, you would not design it like ours. We're stuck with what we inherited from our ancestors. So a lot of our problems with our knees, our hips, our feet, our lower back, it's not ideal, the way we're shaped. We're pretty great. I mean, but we have some things that we could improve. Okay, let's talk about cells. So we're gonna talk about an introduction to cells and how they're studied, and then we're gonna do a tour of a eukaryotic cell. So, do you need to stretch for 1 second? Stand up. Yeah, stand up and assess your bipedal form. Or you don't have to stand up, but you can. I would, yeah, I would. I was thinking about this the other day. How can you sit there and look interested for an hour and a half? I really admire you. So good job. Take a quick breath. All right. Okay, that's all you get. Sorry. So, so let's talk a little bit about cells. Before we get into talking about cells, just refresh my memory. What are the biological levels? What is the most basic biological level? And then molecules, and then organelles, and then well, and then tissues, and then organs, and then and organisms. Very good. So I told you that by the end of last week, you should have memorized that list, right? Which is great that you have. So we're gonna focus on basically these two levels for the next few minutes, okay, or the next little while. All right. So. Okay, so there are different kinds of cells. We've talked about prokaryotic cells, which are basically bacteria. There are many, many, many different kinds of bacteria that live on us, in us, around us. So these three basic types of cells are the defining characteristic of every living thing. So there are three main groups of living things: prokaryotes, bacteria, archaebacteria, which are kind of the outgroup. There are fewer archaebacteria than bacteria, but these are most like ancient living one-celled creatures. So the archaebacteria are a group of living things that are very similar to the earliest life on Earth. They are able to live in very extreme environments. So archaebacteria have been found at the bottom of the ocean, at the edge of sulfuric vents. They can live in very hot temperatures. They've been found deep, deep, deep in the earth, a mile under the crust of the earth. They've been found in clouds, in ice. I mean, they're crazy little creatures. So they're one-celled organisms that are ancient and weird, and we don't need to concern ourselves with them very much. Eukaryotic cells make up the organisms that we are probably most familiar with. So plants, animals, fungi, and a group of one-celled critters, which are called protists, are all eukaryotic. So, protists are important to us in the sense that some of them can cause disease. So for example, malaria is caused by a type of protist, one-celled creature that is injected into the blood with mosquitoes. All right, so we really care about eukaryotic cells. That said, there has been a real shift in thinking, I think, in the last 10 to 15 years about what we are as humans. We have at least 10,000 different species of bacteria that live within us and on us. And you have specific type of bacteria that lives in the creases of the forehead. You have bacteria, it's estimated that maybe 10 pounds of your body weight is composed of bacteria. Now, most of those bacteria live in the gut. So instead of thinking of yourself as one isolated organism, you really should think of yourself as an ecosystem where a lot of stuff depends on you to stay alive, but they also keep you alive. So it's really interesting to think about. And I think that we can't really talk about human biology anymore without talking about the microbiome. How many of you have heard of the microbiome? So the microbiome is a term that describes the bacteria that coexist with us. The microbiome, the bacteria that live with you. So you really are not just a single individual, you are, like I say, an ecosystem. Okay, so in a way, we're really a combination of eukaryotic cells and prokaryotic cells, which is an interesting shift in thinking. So prokaryotic cells, as I mentioned, are smaller than eukaryotic cells. They are simpler than eukaryotic cells. They have DNA as their genetic material, but the DNA is kind of just hanging out inside the cell in a sometimes kind of disorganized fashion in some ways. So a eukaryotic cell has a true nucleus that houses the DNA. So the DNA is only found, well, is mostly found inside the nucleus. The other feature of eukaryotic cells, as we've said, is that they have mitochondria. So they have these organelles. They have other organelles that carry out specific functions of the cell. They're more complicated than prokaryotic cells. A note about size. So cells are measured in a unit which is called micrometers. And the notation for that is a little U with a tail, which is the Greek letter mu. So that's why they're called micrometers. So if you see that little U with a tail and an M following it, it means micrometer. So if you think about this, let's see. This is metric units. Metric. So this is a meter. It's about the height of a doorknob. Meters are divided into 100ths. What is 100th of a meter? A centimeter, right? What is 1,000th of a meter? Kilometer. Right.
So 1 meter is divided into 100 centimeters. 1,000 millimeters. I could do this, 1 million micrometers. Okay, and then I'm not even gonna, I'll screw it up, but a billion. That's an N nanometers. So imagine this cut into a million pieces. That's pretty small, right? So, a micrometer, a typical cell, like a red blood cell, is about 10 micrometers in diameter. 10 micrometers. Can you see that with your naked eye? No. You cannot. It's really interesting. So you require tools to be able to see cells and to see how they are structured. Okay. And the only cell that you could possibly see with your naked eye is an egg cell. Eggs are really big. Okay, they're about the size of a period at the end of a sentence. But other cells we can't see. So this really brings up an interesting point. For most of human history, nobody even knew that there were such things. We're really visual creatures. We don't really believe things that we can't see. And that's partly because we're primates. We really rely on vision as our primary sense, So it wasn't until people developed microscopes that we were able to really understand that we were composed of cells. Okay. So cytology is the study of cells. And it really was possible because of tools that enabled us to see these little teeny things. And the tools are microscopes. The earliest microscopes were invented in the 1600s, and I would expect you to know that. So I want you to know that microscopes were invented in the 1600s. How long ago was that? Approximately. How many? That's really not that long in the grand scheme of things, 400 years. Robert Hooke, who is an Englishman, was one of the people who is renowned for developing these microscopes. This is actually an image from a book that he published where he did drawings of the things that he looked at under the microscope. And this is a drawing of cork. Okay, what is cork? Well, I know it from wine bottles, but part of a tree, right? So he probably was, this is my imagination. So he was probably drinking wine with his little microscope and, shaved off a little piece of the cork and looked at it. But it is part of a tree. It's a plant. And so he saw, so this is dead, right? Dead plant material. And because plant cells, they are eukaryotic, but they have a cell wall that gives them structure. And these are the remnants of the cell walls that he drew, and he called them cells. And that's where the name comes from. So to him, they look like little rooms that a monk might live in. So he called them cells. The other person who's really innovative in terms of developing microscopes was Leeuwenhoek, is his name, Antony van Leeuwenhoek. And he was a Dutchman who, this is one of his very early microscopes, which is still in a museum. And recently scientists have gone and actually looked at materials that he looked at through this little microscope. Leeuwenhoek was very interesting. He was super curious and super excited that he could see these little things. So he looked at everything. He looked at a drop of pond water. How many of you have looked at a drop of pond water under the microscope? It's amazing because if you go out here and get some pond water and drop it on a slide and look at it, you can see all these little teeny things that skitter around on the slide. And he did too, and he called those animalcules. And people said, you are freaking crazy. I mean, insane. He was the first person who had seen all this little teeny stuff. And people really did think he was crazy. And so what he did was he took his microscope on the road to scientific organizations around Europe and said, look, you can see for yourself, these things exist. And that was the first really big realization that there were cells that make up everything that's alive. So this really ushered in the era of microbiology, the study of the microscopic living world. This is a picture that Leeuwenhoek drew of a spinal cord of a cow that he examined under the microscope. This is a very interesting rendering of these would be bundles of neurons that he didn't really know what they were, but he was very good at describing things. So it set the stage for really understanding what all this stuff was. This is looking at red blood cells. RBC stands for red blood cells. So Leeuwenhoek's red blood cells with his microscope versus more up-to-date microscopes, including electron microscopes that were invented in the 1940s that allow us to magnify things by 100,000 times. That's really a lot. So you can see that the detail here is a little bit better than here. So, but he still was able to examine blood and find that there are these little things in the blood. So what we use in the lab, and you'll be looking at tissues, including blood, under the microscope, you'll see something that looks more like this. So again, these are, you can measure these using a microscope. What does this mean? This is a little scale, and this line is 10 micrometers across, and you can see that the blood cells are about that big. This is looking at two different kinds of electron microscopes. So there's a scanning electron microscope that looks at the surface of things like this. And this is a transmission electron microscope that looks at the inside. And what you're looking at here is you're looking at a whole blood vessel, a cross section of a blood vessel. And you're looking at cross sections of red blood cells within the blood vessel. So it gives you an amazing view. There are lots of techniques that have been developed in the last 10 years to even improve our vision, including, so this is looking at a mouse cell using a technique called fluorescent microscopy, and it's where you use bioluminescent proteins from creatures who have that ability to light themselves up. You can use those genes and attach the proteins to certain structures that you want to highlight, and then you view them with this special microscope called a fluorescent microscope, and these things then become even more apparent to you. So this bright green tangle of fibers represents a certain kind of structure inside our eukaryotic cells, which is called microtubules. Microtubules form part of the internal skeleton of the cell. It's a framework, a protein framework that maintains the shape of the cells and also provides little pathways like railroad tracks that you can move material from one part of the cell to the other. It's truly amazing. So the kinds of details that we can see now have given us much more insight into the structure and function of cells. Plus, they're beautiful. So we have a lot to learn, but we have learned a lot already. So the earliest microscopes were developed in the 1600s, and they became better as the decades went on. And in the 1800s, by the 1800s, microscopes had been developed that are very similar to the ones that you may have used in labs, where you have the ability to magnify things up to about 1,000 times. And so these new microscopes allowed scientists to ask more sophisticated questions about the cellular level. Okay, so this was a time in the 1800s when a very important concept was articulated. And by articulation in this sense, I mean, it was written down and told to people. So that concept is the cell theory. And I would want you to be able to explain the cell theory. So what is the cell theory? It is a major concept of biology. It's the idea or the observation that all living things are composed of cells. All living creatures are made of cells. Whether you are a plant or an animal or a bacteria, all living things are built out of cells. That was elaborated a little bit to say that not only are all living things composed of cells, but all cells come from other cells. You don't just appear out of thin air. This may seem sort of intuitive to us today, but in the 1800s, it was not. So in the 1800s, there were some very important, two important scientists, Schleiden and Schwann, who came together they collaborated to publicize this idea. So Schleiden was a plant biologist. He was studying plants, and Schwann was studying animals. They took samples from their organisms and they were looking at them under the microscopes, and they began to talk to one another. And Schleiden said, you know, all the plants I look at, they're made out of these structures, which are cells. And Schwann said, hey, all the animals that I'm looking at, they're also made out of cells. So that's why they came together, and in the 1830s, they published a paper that was the cell theory. All living things are made out of cells. Wow, okay. So that's groovy. That's important. So we now have the ability to do a lot with cells and understand them much more detail. So 1839 was a long time ago, right? Almost 200 years ago. So in the past 200 years, we've made some progress in our understanding of cells. We can now grow cells in the lab. That's called culturing cells. We can culture cells in the lab. It simply means that you grow them. Cells are, those cultures of cells that are grown in laboratories are now used all the time to test drugs, okay, to determine the effect of toxins, to assess treatments, pharmaceutical treatments, to figure out processes of life, how the cells work. So these particular cells are muscle stem cells that have been grown in the laboratory. And they have been used, for example, to treat mice with a condition that is similar to muscular dystrophy. So we can use cells to help us address diseases. So, you know, they're planning to culture animal cells to grow meat in labs. That's already being done. So, do we need to stand up for a minute? Absolutely. Go ahead and stand up for 1 second. Okay. So we'll see you in a minute. Okay, so let's go on. So current updates to the cell theory, some big ideas about the cell theory. The cell is the simplest structure to carry out all life functions. An organism's structure and function is ultimately due to cellular activity. So really, we can only understand health and disease if we understand the cellular level of what's happening. Something that Schwann and Schleiden couldn't have known when they developed this idea in the 1830s is that we can trace all life back to original cells that lived about 4 billion years ago. For one thing, they didn't really have a coherent concept of evolution. So it wasn't until 1859 that Darwin wrote The Origin of Species. Charles Darwin was really the person who clarified what evolution was and how it worked. So that was after Schwann and Schleiden. With our current techniques for comparing DNA of organisms, we know that all living things share DNA. And we can even create the trees of life that I shared with you that showed that the earliest living things were about 4 billion years ago. The other thing that Schwann and Schleiden couldn't have had any idea about is the chemical composition of cells and their metabolic processes. So we all, as living things, have two main things in common. Protein synthesis and extraction of energy from the environment to produce ATP. Those are two aspects of what are called metabolism. You probably have ideas about what metabolism means, but the basic meaning of metabolism is all chemical reactions that occur in a cell. So all the chemical processes that occur in a cell. The two main ones are protein synthesis and extraction of energy from the environment to produce ATP. So what we now know, again, that Schwann and Schleiden couldn't have, is that the cells, whether you're 4 billion years old or a prokaryote or whatever, we all are chemically, our cells have the same chemical components. And they perform a lot of the same activities. So all living things are actually very unified. All living things are unified because of our reliance on DNA and because of the way that our cells work. I can talk about this sort of thing forever, but I don't want to necessarily keep you forever. However, we are gonna go on because we have so much to cover. We're gonna go on to a second part of the cellular discussion, which is just beginning the discussion of major parts of the cell. So 4 major components of the cell that we're going to be discussing are the plasma membrane, which is sometimes also called the cell membrane, okay, cytoplasm organelles, and the cytoskeleton. Okay, so what you will be able to do is define a stem cell and be able to define the term differentiation. And then you'll be able to give a basic definition of eukaryotic and prokaryotic cells. And you will be able to describe the plasma membrane and some of its functions. Remember that these PowerPoints are available to you, and this is basically your study guide. All right, so these are in the modules. So this is a common kind of depiction of a eukaryotic cell. So it would be a specific type of cell in a multicellular organism. This type of cell that is depicted in this picture is an epithelial cell. So remember, an epithelium is what? That term, one of the types of tissues, okay? So remember that you as a human are made up of about 200 different kinds of cells. So not all cells look like this. You have muscle cells, you have nerve cells, you have bone cells that don't look quite like this. It's kind of a generalized epithelial cell that you might find lining the tubes of the body, such as the kidney, where your urine is produced, or lining part of the respiratory tract or the digestive tract. So what this cell depicts that all eukaryotic cells have, whether it's a neuron or a bone cell or what have you, is the nucleus. And this is depicting the nucleus. The cytoplasm is the material between the nucleus and the plasma membrane. So the plasma membrane is the envelope that encloses the entire cell. You with me? You guys are doing good. You're hanging in. Okay, so the plasma membrane encloses the entire cell. Why do we call it a plasma membrane instead of simply a cell membrane? Because these organelles are mostly also enclosed in membranes. So these would all be cell membranes, the envelopes that include the specific organelles. So plasma membrane specifically is a term that means the outer envelope of the cell. Okay. Do you see the difference? Cell membrane is more general. Plasma membrane is just the outer envelope. So pretty incredibly, and I've mentioned this several times, we start as a single cell. So as mammals, there are biologically, there are sperm cells that come from males and an egg cell that comes from a female. I mentioned to you the egg cell is enormous. It's got a lot of cytoplasm because the nourishment for the developing critter for the first part of right after fertilization for several days, the nourishment comes from the cytoplasm of that cell. So this is the enormous egg cell. During fertilization, well, I mean, a male, I know it's kind of gross to think about, but and well, No, now I'm gonna blush. Damn it, I hate that. But a male produces about 200 million sperm during ejaculation. And only about 10,000 of those get anywhere near the egg. So it's a treacherous path up there. They get stuck, they can't swim, whatever their problem is. And then there are two ovaries in a mammalian female, human female. So one ovary each month pops out an egg. And so there's a right and a left path, so only some of the sperm go up the right side. So about 10,000 of them get to the vicinity. And it's truly amazing because they get closer, some of them approach the egg, and the egg has proteins that allow one of the sperm to penetrate. So one of the sperm gets in and then the egg closes the gates. So the membrane becomes impenetrable. And there are enzymes that exclude the other sperm in that area. So only one of them usually gets in. And so this is the mass of sperm around the egg. Once the sperm enters into the egg, it drops its tail. And basically what the sperm donates is the nucleus. So the whole point is to get the DNA from the male to combine with the DNA from the female. So they both contribute 23 chromosomes to this developing conceptus, it is called, which is this fertilized egg is called a zygote. So usually fertilization in humans occurs up in the fallopian tube, close to the ovary, and then the zygote begins to float down the tube toward the uterus. And as it does so, it begins to divide. It doesn't grow in size, but the number of cells grows. So it does this within the first 72 hours after fertilization. It's a ball of cells that develops. It comes down the fallopian tube, and then about a week after fertilization, you now have a hollow ball of cells that implants on the wall of the uterus. Okay, so it's crazy, 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 crazy. So we'll talk more about this next time. Remember, Thursday, there is no lecture. So Thursday, what you can do this week is complete your assignments in Connect. And you can read through the modules. Okay, so there's a module on the cell. There are some videos that I want you to watch. Okay, short and fun. All right, thank you guys so much. And we'll see you, if you're in lab on Thursday, I'll see you on Thursday. I'll see you otherwise next Tuesday. Okay, awesome. Let me,